you've built this autonomous lab designed for machine to machine, or you're building this autonomous lab designed for machine to machine operation, and you've got this collaboration with OpenAI. Can you talk a little bit about the work you're doing? And I'm really interested in like what a lab in the loop architecture looks like.
Yeah, this is really neat. You can come by our Seaport facility. This did not necessarily exist this level when Datapoints started. We have a lot of different automation throughout Ginkgo. One piece of automation that we're investing heavily in, though, is a unit called a rack, a reconfigurable automation cart. We've actually been developing this internally for many years now. It was actually started at Zymergen, and we acquired Zymergen. So it has about 7 or 8 years of product development to it. And it really finally reached the point now, and I think that the field is actually ready for it, in that we're establishing these large system like autonomous labs that are able to generate scientific data, not just at high throughput. We've seen that before with work cells. That's some of the technology that Datapoints uses from other kind of automation providers. That's not so new. What's new about the autonomous lab is that we have 100 different instruments on one system, and it's really meant for diverse data generation, right? And so it can do more than one protocol at a time. 80, 100 protocols at a time. And it really is replicating what is happening in the lab today from a physical architecture perspective. That is, if you go into a lab, you see this linear layout where you have these black bench tops with instruments on each, on each section of it. And then you have a human that actually goes about and transfers samples from instrument to instrument in the order that they need it and at the time points that they need to do it. This replicates that. It is a linear setup. However, it is a magnetic rail that transfers the plates from one instrument to the next. And instead of a human dictating when that should happen, it's, of course, a human writing the protocol. But actually, you have then the Catalyst software that we have driving where the plates need to go at a specific time. And it's orchestrating all these in real time so you can have multiple protocols at once. Now, I mentioned, and I said, of course, it's a human designing the protocol, but that's not always true. And so we did a really neat collaboration with OpenAI where they used GPT-5 to actually design experiments to optimize the output of a cell-free protein expression system. And so this is really important. Cell-free protein expression systems, simply speaking, are you take away the cell membranes, you have the rudimentary mix of what you need in order to translate proteins and make proteins. And you're able to do this with a number of components, but those components need to be optimized. The ratios of those components, which ones you add, are there any that you can remove? This is a big multi-parameter problem. And of course, there are consequences to what your recipe is. That is, you can not only have higher or lower titers, but there's also a cost perspective. And so the goal for the industry, and cell-free protein expression is used in some drug development and actually commercially approved drugs. It's also used in other industrial applications. But the idea is that you get a higher titer of protein expression from each volume of lysate that you use. And then you have a lower cost point. That's the goal, right? So more protein, less cost. We did about 36,000, 40,000 different reaction conditions using GPT-5 to actually select those reaction conditions. What was amazing about this is that GPT-5 was writing its own ELN. And so we often talk about what is the interpretability of AI models. In this case, you actually had the LLM telling us exactly why it chose to do what it did. And it also was looking for new reagents. Like, what are some reagents that we haven't thought about before? And they actually suggested some pretty unique ones. Some of them we couldn't actually source. There is a human component to actually doing procurement for the reagents and loading the reagents onto the system. But for a large part of this, GPT-5 was actually designing the experiments and the protocols, and then the autonomous lab that we have, what we call Nebula, based on these rack setups, reconfigurable automation carts, they were really doing the experimentation. And I think this is a really neat example of what can be done with autonomous labs. Now, it's not just a GPT-5 that can do this. The humans at Ginkgo do this. We also have a cloud lab interface where other scientists can actually access this and submit samples as well. And there's even some sense that maybe you could have a bake-off one day, almost like the the Go type of competition where our cell-free protein expression did reach a new goal. We successfully had the lowest price point per titer in the industry that has ever been reported before, and GPT-5 did that. There are some academics here. In fact, one that probably was influential in the way that GPT-5 learned, what some of the best experiments could be. Michael Jewett from Stanford, who is an expert in the space. Maybe one day we could actually see him, and you actually have a human that's designing experiments. And if he had the horsepower of Nebula behind him, perhaps he could even come up with a better outcome. So I think this is a really interesting way to think about where the field of autonomous lab is going, and the role of humans in that loop.